Hello, this is Mahanta Didia. This is the third related video of the second module of this book. This is the first book on liquid formulation that utilizes qubits video format, covering both industrial pharmacy and cosmetic science. This is an on demand, dynamic supplemental book. The innovative pharmaceutical and cosmetic companies build quality in product development. In the later modules and related video, we will include examples of quality by design used in solution, suspension, and emulsion formulations. In this submodule on physicochemical characterization of solution dosage form, quality by design concept is introduced. In addition to the example of quality control specification, stability of formulation, and chemical kinetics of active moiety are presented. Quality, stability, and chemical kinetics. Product quality. Quality of each product, especially in pharmaceutical and cosmetic, is ensured by three distinct elements: quality by design, quality control, that's a testing of ingredients and finished product in laboratory environment, and stability studies conducted to establish shelf life. Quality by design concept was introduced about 20 years ago. This looks at critical material attributes, CMA. These are physical chemical property of ingredients, critical process parameters associated with manufacturing of the product. These are mixing conditions, temperatures, and related parameters. Critical quality attributes are quality standards and risks. Each CQA is assigned a risk, low, medium, or high, of meeting the quality standards. Based on CMA, CPP plan, and the control strategy are designed to lower or mitigate the risk it poses on quality of products. For example, in earlier submodule, we looked at precipitation of proteins in presence of salt. Similarly, preservatives can precipitate at lower temperature. Critical material attribute, CMA of preservative, and the storage temperature. QBD, quality by design, must be built during product development and continuously monitored in production through distribution. It starts with defining the product profile, product design understanding, formulation, process design understanding, develop control strategy, quality controls, and manufacturing and CMA strategy, and continuously improve. This is a requirement for pharmaceutical. In this slide, it's a very busy. However, it's important to see in totality what CQA, CMA, CPP, and its effects are. Particle size, globule size, or clarity of the solution depends on material properties, material particle size. In a manufacturing mode, it depends on multiple things, including temperature and mixing conditions. If these are not controlled, then it will result into content uniformity, release of the drug, and patient compliance will suffer due to attributes of the product. Rheology, these properties depend on material attributes, viscosity of the material, as well as manufacturing conditions. The effect of this is how much material is retained on the skin and how much is permeates. What are the sensorial effects of the product? If the viscosity or yield stress are different, then patient or a person will feel that. Evaporation of or solvents, that will change the composition. For example, if water were to evaporate it was precipitating or crystallized solutes. It would depend on manufacturing conditions such as process temperature, container closure systems, mixing conditions. How homogeneous product is depends on a ingredient and its properties, and also on processing. If the materials are not homogeneous, it will affect skin penetration and therapy. Precipitation and aggregation depends on solvent type, co-solvent type of emulsifier. It depends on manufacturing conditions. And the effect would be active pharmaceutical ingredients or active drug will precipitate, and that will not be acceptable. Microbial limits, contaminated materials in humans, lack of cleaning protocols. We need certain amount of microbial agents to protect the product. These CQA must be addressed during development and have to be continuously monitored. Example of QBD is shown here. In each of the module on solution, suspension, and emulsion, QBD examples are shown. In this case, to introduce the risk assessment, quality attributes are shown for a solution of fenfluramine hydrochloride. This drug was reviewed by FDA, and they identified several CQA. One of them was dosing accuracy. It was assigned a low risk. However, risk mitigation approach was viscosity and delivered volume be provided in quality control specification. In addition to that, pharmacy to apply a syringe. Palatability was identified for fenfluramine as an issue, initial risk being medium. This risk was then defined as adequately mitigated. Leachable from plastic containers was identified as a medium risk, and again, study data was provided. So quality attributes is different than quality control. Quality control is a test method and specifications. Example here is for Theophylline oral solution. It states that it needs to be between 95 to 105% throughout its shelf life. Identification methods are prescribed in detail in USP monogram. One is thin layer chromatographic identification system. Second is a retention time of a major peak through assay procedure. Details are given of the chromatographic system as well as calculations and acceptance criteria. Additional testing, the microbial test, pH, product contain alcohol determination needs to be made. And it provides additional requirement of this is a quality control specification. Three components of quality are quality by design, quality control, and stability. Stable product must maintain its properties throughout the shelf life at specified level. It must meet quality control spec throughout the shelf life. Stability studies are conducted in a container to study the influence of temperature, humidity, and light for a specified time period. In addition to that, establish anticipated shelf life of a drug product and recommend storage condition by use of chemical kinetics is physicochemical characterization of the product. And in this submodule, we'll look at this with more detail. Physical stability and package integrity includes physical appearances, particle size, solubility, pH. These parameters can affect shelf life. Microbiological stability is conducted to establish microbial limit testing. These are included in quality control specifications. Chemical stability and kinetics is studied as part of physicochemical characterization. Example shown here is methyl and propylparaben. Ester link here can degrade and it would generate parahydroxybenzoic acid. which further can degrade into benzoic acid and phenol, and should be able to predict activity of parabens based on kinetic studies so that we can establish shelf life specification. There are three major reaction rate. Degradation reaction can be a zero order that's independent of the concentration of the ingredients. More commonly is a first order reaction in which reaction rate here, the degradation rate of paraben into p-hydroxybenzoic acid, for example, is proportional to the concentration of the parabens or active ingredient. Complex multiple orders in some cases where rate of reaction is proportional to the multiple powers of the reactant. Calculate shelf life. First order reaction rate is given by rate is proportional to the concentration A, where rate is in the terms of mass per time and K is the reaction rate coefficient. Its units are per time. And this K is calculated to establish shelf life. A is an amount or could be concentration. If we integrate this equation, natural log of concentration at any given time is equal to the natural log at time 0 minus K times T. Minus sign indicates its degradation. This can be also written in terms of concentration. In addition to that, reaction rate changes with temperature. It is described by an Arrhenius equation. K is equal to A E activation energy divided by gas constant times the temperature in Kelvin. The reaction rate K is inversely proportional to temperature in Kelvin. K can be calculated from a graphical mode. We can measure either amount or concentration or several months or several days and graph it to show that concentration for first 48 months here. First order as shown in this equation with R square of 1. Degradation rate coefficient K and shelf life. Stability studies are performed according to regulatory and industrial standards to establish expiration and shelf life. Acceptable criteria 5 to 10% degradation of active, and all degraded meet toxicological threshold. Shelf life of more than 24 months at storage temperature is desirable. Based on accelerated condition, we like to establish will product meet 24 months shelf life at storage temperature. This can be done using Arrhenius equation. We will see that in the next slide. Long-term and accelerated condition. Long-term stabilities are defined as expiration date at storage condition, for example, 25 degrees at 60% RH, what's called room temperature, or in refrigerated conditions such as 2 to 8 degrees. Accelerated stability is at a higher temperature such as 40 degrees, 75% humidity for 6 months. Degradation kinetics are conducted at high temperature and very short duration. It utilizes Arrhenius equation K equal to A E minus activation energy divided by RT. And we use that to project the shelf life. Actual establishment of the shelf life has to be done using long-term stability. However, we need to project the shelf life to decide what conditions will be the best. This can be done with degradation kinetics, generally done at 50 degrees, 60 degrees, 70 degrees for very short duration. At each condition, Degradation kinetic experiment is conducted. Product is exposed, for example, at 50 degrees for 21 days. The samples are taken at different time intervals to see how much active has degraded. Then we graph natural log of concentration versus time, calculate K value at different temperature conditions. We calculate K values and then graph Arrhenius plot. Based on Arrhenius plot, we can calculate K value at the room temperature or desired storage temperature and the projected shelf life. Degradation of ascorbic acid in sugar syrup is shown here. Degradation is determined by kinetic experiments at different temperatures: 32 degrees, 45, 60, 70, and 80. And they would be exposed to very short duration. Studies are conducted for short duration. Based on the concentration, the slope of the curve is determined. Slope is negative, indicating degradation. 80 degrees study was conducted for 9 days, and degradation K value was calculated as 0.11650. Similarly, 70, 60, 45, and 32 degree studies were done. 32 degrees, for example, was done for 20 days and was 0.00258. This data now can be used in a radius equation. A radius equation was shown earlier. It is a relationship between natural log of K versus 1 over T. We know the value of K from previous slide. We can calculate absolute or Kelvin temperature, 273 plus temperature in Celsius. We can calculate 1 upon T and natural log of K. So for example, 60 degrees, degradation rate is 0.02285. Temperature Kelvin is 333. 1 upon 333 is shown here, and natural log of 0.02285 is shown here. Natural log of K versus 20. And this was done with an Excel software, and with Excel, linear trend line and equation was projected. This can be done manually using a log paper also. So the equation for ln K is minus 9712.2 multiplied by 1 upon T plus intercept of 23.875. Once we know that, we can plug in the value of T, in this case 25 degrees, and substitute T at 298, we can get the value of at 25 degrees C of 0.001004. So this is a shelf life projection based on 90% of the active be present during the shelf life. We can calculate shelf life as 105 days at room temperature. This kind of physicochemical characterization provides information about the quality of the product.